So in this lecture presentation, we're going to be covering the urinary system. You guys got the share. Okay, this one's funny. There aren't many funny human anatomy cartoons, but this one kind of cracked me up a bit. Okay, so in your Blackboard learning module, you went over the structure of the nephron. So in this presentation, we're going to cover some of the functions. So urine formation consists of three main processes. The first is filtration. Now there's a lot of like extra words that are surrounding this terminology, so I'll try to break it down. So filtration is separation of molecules or substances based on size. So even if you had like a sieve or a colander and you wanted to screen dirt to remove rocks, if you put this dirt in a sieve, the rocks would remain behind and the dirt would go through. So that's like the same exact type process. So your blood enters into the glomerulus, which contains very leaky type of capillaries. Think of a garden hose that has a bunch of nail holes punched into it. And so this is going to filter out all small molecules. And it's not urine yet, so it's referred to as filtrate until we get to the last stage. So the filtrate's going to contain, again, small molecules such as water, electrolytes, glucose, urea, uric acid. The filtration or these filtration slits are too small to allow red blood cells, white blood cells, platelets, blood proteins to exit. So you just have a filtrate containing very small molecules. Okay, however, you don't want to eliminate all these small molecules. So urine is going to contain predominantly water, help balance out your overall water balance within your body. It's going to contain some waste products, but most of the water that enters into the filtrate needs to be reabsorbed. So if something's reabsorbed, it's going from the tube back into your bloodstream. Okay, so this is called tubular reabsorption. So you're reabsorbing water into the bloodstream. And the area of the nephron where this occurs is proximal convoluted tubule. So convoluted doesn't look really twisty here in this picture, but it's like a twisted, turned area of the nephron. And proximal means that it's closest to the glomerulus and the glomerular capsule. Okay, if I didn't mention, when the filtrate is initially generated, it's kind of trapped in the capsule. You can see that surrounding the glomerulus, and then it continues to the proximal convoluted tubule. Later on, we'll discuss the distal tubule, which is further away from the glomerulus, right? So proximal is closer to the source of origin, and distal is further away. Okay, this is going to reabsorb about 65% of water and amino acids, electrolytes, and glucose, because these are substances that your body needs. To me, it always sounds counterproductive. You're filtering all this material and then you're reabsorbing most of it. But in the human body, there's really only so many ways that you can get water to go from one area to the other, or in addition, other small substances. There's only so many ways you can move molecules, right? So you have to kind of filter it and then reabsorb what is still needed. Okay, in the nephron or renal tubule, so you can see there's the renal tubule, kind of dips down. Okay, it goes across kind of a hairpin turn and then comes back up. So they kind of call this descending limb, okay? And then there's the hairpin turn and then ascending limb. So it's going down, over, up. This is where you can continue to have water reabsorption. Only water is reabsorbed in this area. So if you had, say, if somebody's a diabetic and they have an elevated blood glucose level, they are not going to be able to reabsorb all the glucose that enters into the proximal tubule. So the rest of the glucose is going to travel along, go through the renal tubule, and then end up ultimately within the urine, right? So a screening test for diabetes mellitus is looking for the presence of glucose in someone's urine. Here, there was my grandfather's clock in the background. All right. So the amount of filtrate that's generated in a day is about 180 liters. So think of like a liter or 2 liter bottle of soda. That's a lot of water. Obviously, you're not urinating that much. You're reabsorbing most of it. Average urine volume per day is between 0.6 and 2.5 liters. So that's how much is actually being reabsorbed. The amount of urine produced is going to vary by like how much water you've had to drink, the food you consumed that were very heavily like water based. Have you been exercising? Have you been sweating? Like, what are the environmental conditions? So that's why there's kind of a wide range on normal urine production. Okay, now we're going to the distal convoluted tubule that is going to connect to the collecting duct. There'll be many, many nephrons attached to one collecting duct. And this is where you do kind of like your last fine tuning. So you might be, secreting acids or bases, particular ions. I'm not going to go into a lot of detail about this, but you're kind of doing an acid-base balance type of molecular movement. What we're going to kind of focus on is normally this area is impermeable to water. So you're not going to have any water resorption. You will have water resorption, but it's under hormonal control. Okay. So this would be a situation where you haven't had a lot to drink or you've been exercising and sweating. And when your, water balance starts to drop, right? That is detected in the hypothalamus. And then kind of another regulatory mechanism is if your water balance goes low, it drops your blood pressure. We're going to get to these later. All right, so we mentioned like the acid-base balance, and this, when you have substances that are going from your blood directly into the tubule, this is called tubular secretion. So this is the reverse of reabsorption, right? And this actually occurs in the proximal convoluted tubule. So we mentioned, right, proximal convoluted tubule, that's where reabsorption of water, glucose, and amino acids. To me, this is like a shortcut way of getting rid of substances that perhaps not enough of them were filtered out in the glomerulus. So this could be like hydrogen ions, ammonium ions, potassium ions. You don't have to worry about all the names of the ions. Creatinine, this is a kind of metabolic product of muscle metabolism, some hormones. We'll kind of just talk about maybe the last one listed because it's probably like the easier one to remember. You have some drugs that are regularly secreted into your urine. And typically, these are drugs that you need to take several times a day, or now a lot of pharmaceutical manufacturers have drugs that have like a slow release, right? So if you take penicillin, it's very quickly secreted and removed from your body. So this would be like a type of antibiotic you have to take like 3 times a day or 4 times a day. And another example is beta blockers. But that's maybe interesting thing to remember. Okay, you don't need to know any of the numbers on this table, but if you just look at it, you can tell whether something is being reabsorbed or being secreted, right? So if you look at all these numbers, your plasma level and your filtrate levels are about the same. And then you have the urine levels. So if you look at sodium, if your filtrate level is 142, and it's like milliequivalents per liter, right? And then your urine level is 126. Obviously, you reabsorbed some of the sodium, right? If you look at potassium, right, your filtrate levels is 5, your urine levels are 60. So obviously you secreted some potassium. So it just kind of gives you an idea which molecules are going which way. Now, often, again, we don't have time in this course to go into all of the mechanisms that are involved here, but the urinary system, the number one like rule to remember is water always follows the salt. Okay, so salt being sodium. Water is kind of difficult to get to move from one area to the other. It's kind of difficult to get it to move from inside the tubule to back in the bloodstream. But if you reabsorb sodium, that sets up a concentration gradient that water would then tend to follow. Water is going to always go from where it has a high concentration to a low concentration. So if you reabsorb a bunch of sodium, right, you're making that area more concentrated in sodium, which means it has a lesser concentration of water, and then the water will kind of naturally follow along. All right, so let's get to the distal convoluted tubule. And then that, again, many nephrons are going to be attached to the collecting duct. And normally they're impermeable to water. And so whatever's in the filtrate, once it gets through the nephron or renal loop, is going to go into your urine. But if anti-diuretic hormone, ADH, is present, and the way I remember this is, all right, if you take a diuretic, it makes you urinate, right? It makes you pee more. So anti-diuretic makes you pee less, okay? So what this does is it makes these segments permeable to water. Water is reabsorbed, and then your urine is going to become more concentrated. Okay, we'll get to the mechanism of this shortly. Okay, this one's kind of funny. Got to make sure I'm sharing the right screen. This is good. Looks like I mean, hang on. Hang on. Okay, there we go. All right, so we're not gonna really worry about the aquaporons, but what they are, let me kind of go back to diagram here. All right, so in the proximal tubule, in the distal tubule, I get a good picture if I go forward. Sorry, guys, I'm trying to find a diagram to explain this. We have to go back to the beginning a bit. Yeah, this one's pretty good. And I know this is discussing tubular secretion, but it does show you where the distal tubule would be in the collecting duct. And so it's normally impermeable unless these little proteins are inserted into the cell membranes forming the tubule, the aquaporins, and then that allows water to exit. Okay, so don't have to worry about the term aquaporin, but just know that when ADH is present, that allows for water reabsorption from the distal convoluted tubule and collecting duct. Okay, right. So this is how this works. All right. So if you were starting to become dehydrated, right? The concentration of the water in your blood decreases, right? So you have less water in your blood. And so this increases the osmotic pressure. So this is a measurement of the solute concentration, right? So your blood is really a solution. It consists of two things. It consists of the solvent, which is water, and all the other molecules, which are solutes, right? So when your water levels, percentage wise, your water levels go down, that means the solute concentration goes up, and that's an increase in osmotic pressure, right? So make that a little easier to understand. So you have it, if you have a decrease in water, that means you have an increase in osmotic pressure, right? So there's osmoreceptors in the hypothalamus. Remember, hypothalamus is the kind of key regulatory center for homeostasis, and then it triggers the release of antidiuretic hormone that is then carried to the kidneys, and it causes the distal tubules and collecting ducts to increase water reabsorption. Don't worry about the term osmosis. Osmosis is just a diffusion of water, right? So it allows water to reabsorb and therefore makes your urine more concentrated and decreases your urine volume. Now, there is another mechanism that increases water reabsorption in this area, and this is in response to a drop in blood pressure, right? So aldosterone is secreted by your adrenal gland due to a drop in blood pressure entering the kidney, right? So the kidneys and the heart sometimes battle with each other a little bit, right? So your heart, your cardiovascular system doesn't want your blood pressure to drop, okay? So this sometimes then may cause your kidneys to have to work a little bit harder, right? So there's a little bit of a like a give and take here between the kidneys and the heart. So your blood pressure drops, and this could be, for instance, if you are hemorrhaging, or it could be other factors. Um, it could also be triggered by dehydration. And so this is going to increase water reabsorption in the same areas, distal convoluted tubule. It's just the mechanism is slightly different. So it's going to increase the reabsorption of sodium ions. And we said rule of urinary system is water follows the salt. So sodium ions are reabsorbed, and then the water is going to follow. And that's going to increase your blood pressure by increasing your blood volume. So the higher your blood volume, the higher your blood pressure. If your low blood pressure is fine if you remain conscious, but if it gets too low, you could suffer from hypervolemia and go into shock. So your blood pressure can drop too low. Right? Can you drink too much water? Answer is yes. It says, and you die. Yeah, so this is interesting because these are like parts that we've studied previously and even in this unit. So it's saying your brain starts to swell, it starts to push through the foramen magnum, which we just covered in lab last week, and then crushes your medulla oblongata. So that controls a lot of vital functions, and this can lead to death. So in this case, it's you would never be able to do this accidentally. This is something that you deliberately would have to drink that much water in a very short period of time. And honestly, I think most people would really become too nauseous to be able to even continue. Okay, so another function of the kidneys, which is kind of totally unrelated to water balance, is when blood flowing through the kidneys. If it contains low blood oxygen levels, the kidneys release erythropoietin, and erythropoietin is a hormone that stimulates the production of new red blood cells. So remember from the cardiovascular system that your red blood cells are very short lived. They only live about 120 days. And so, as red blood cells are becoming old and fragile and are getting destroyed, that's going to cause this little bit of decrease in your blood oxygen levels. And this hormone then helps to stabilize your overall, quantity of red blood cells. Right? So it's released, then your blood oxygen level stabilized, and then it's not released. So it's going back to first class. This is an example of negative feedback. Right? So after you've gone through the glomerulus, you have the filtrate, you have reabsorption and secretion in the proximal tubule, and you have only water reabsorption in the um, renal loop or nephron loop, the part that goes down and up. And then you may or may not have some reabsorption of water in the distal tubule or collecting duct. Okay, just to kind of summarize everything we've done. Once it's in the collecting duct, it's then considered to be urine. Okay, so urine is that 95% water, usually contains um, waste products of metabolic activity such as urea, uric acid, creatinine, may have some trace amounts of amino acids. Again, just electrolytes, that's how this acid-base balance occurs. Okay, I'm not going to worry about the names of all the different electrolytes. And your volume of urine, we said, again, average volume 0.6 to 2.5 liters. It's going to vary with how much fluid you've been drinking and environmental factors. For instance, how hot it is outside, how much you've been exercising or moving around. You should not have any glucose, proteins, ketones. Ketones are metabolic products of fat metabolism, or blood cells. They should not be found in urine, and their presence may indicate disease. Like I mentioned previously, glucose found in urine is indicative of diabetes, diabetes mellitus, full name. But if you just like pigged out on all your Halloween candy, so did that. But if you did that, you may end up with some trace amounts of glucose there. So urinalysis, excuse me, urinalysis is kind of a screening procedure. Having glucose, proteins, ketones may be based on something you ate, but you should not ever have red blood cells or white blood cells unless you have some type of infection. So urine, if you looked at a normal urine specimen, I tried drinking some water. The pH, the average is about 6. There's kind of a wide variation depending on your acid-base balance at that particular time. Specific gravity is like a measurement of density. So water has a specific gravity of 1.000. So any substance, like any solute, such as urea, uric acid, creatinine, added to water, in this case your urine, is going to increase this volume. Why is this important? If your specific gravity is too low, it means your body is not releasing or not allowing some of these substances, such as urea, uric acid, to enter into your blood. And these contain nitrogen, and they're a waste product that really needs to be eliminated. So some blood tests that are done, like are your blood urea nitrogen level or your creatinine levels. So if your blood levels are too high, that can be indicative of kidney disease. Okay, so too low is bad. Could be kidney disease. Too high means there are kind of like too many solutes are there. And this could include, for instance, like glucose, right, proteins, or ketone bodies. Right, 95% water. We've mentioned the average volume per day. Your urine is kind of a clear yellow, and this is based on the presence of the pigment urochrome, which actually is a breakdown product of hemoglobin metabolism. And the color may vary sometimes, like depending on like what foods you ate. Um, Obviously, if you're on the lower end, like the 0.6 liters, it's going to be a little bit darker. If your urine is, if you urinate more frequently, if you're drinking a lot of water, it's going to be a little bit lighter. The odor can vary. That depends on like maybe foods that you've ingested. So one thing like asparagus is a food that can cause a very strong odor to your urine, and it should be sterile. So we're lucky today that it's very easy to do all these tests. Um, Historically, physicians would actually like seriously, it's like a glass of wine. Swirl your urine, look if it's cloudy, make sure it's clear, that it's yellow. They had to smell a patient's urine and they would actually taste the urine. So if it had a kind of sweet taste, then that would indicate diabetes. Yeah, things have definitely progressed in that area of medicine. All right. So once we have this filtrate that's formed, it's going to go into the collecting duct. Remember, there's many, many nephrons attached to a collecting duct. And then it goes to the renal papilla, which kind of just means like little bump. And then it's going to pass into the minor and then major calyxes, which we looked at in lab. And then it continues to the renal pelvis. Remember, there's three regions of the kidney. There's the cortex, medulla, and pelvis. And from the pelvis, it's then going to leave the kidney by the ureter. The ureter is going to transport the urine to the urinary bladder for storage, and then it exits the body through the urethra. So the ureters, again, kind of connecting the kidneys to the bladder. And it doesn't go into the top of the bladder, which, would kind of make sense. But the bladder, it can expand when it's full of urine and then it collapses when it's empty. So the tubes actually go into the bottom part or the base of the bladder. So this urine is kind of constantly leaving the kidneys, going through the ureters into the bladder. And so peristalsis, which we studied with the digestive system, it's rhythmic contraction of smooth muscle that moves substances along. So there's peristaltic waves and gravity. This assists in moving urine into the urinary bladder. So there's just a little valve like a flap like valve, and that allows urine to enter but not to exit once it's gone into the bladder. So where everybody knows this, right? You can be laying down sleeping all night, and so you're not really having gravity assist the movement of urine, but then the peristaltic waves will assist it. So if you're ever, I don't know, you're ever like sitting down and you're like, yeah, I think I need to urinate. And you stand up, you're like, yeah, ooh, I really feel like urinate. So gravity is an assister in its movement. Your luck. Sorry. So this is just kind of showing a cross section of the ureter, and this is also like expandable depending on how much urine is passing through it. So the bladder, it's a kind of a hollow muscular organ. It can distend or get bigger, right? It's in the pelvic cavity, and it can store between 200 and 800 milliliters of urine. So it stores the urine, which is then going to exit through the urethra. So the urethra also exits from the base or the bottom of the bladder. So the floor of the bladder has this area called the trigone. Just think triangle. It's just the openings for the two ureters and the one urethra. So this is showing urinary bladder in a male, and you can tell it's a male because the prostate gland is present, and it kind of shows that triangle or trigone at the bottom. Right, the picture on the right, you can see the ureters and how they're exiting the bottom part of the bladder. The urethra is the tube that transports urine from the bladder to the outside of the body. And similar to what we discussed with that digestive system, it has two sphincter muscles, round muscles that control the movement of urine from one area to the other. So you have the internal urethral sphincter, which is under involuntary control, and the external urethral sphincter, which is under voluntary control. So remember, smooth muscles are involuntary control, skeletal muscles are voluntary control. So micturition is when the urine leaves the bladder, and this is by a reflex. So let's see if I can get the video. It's an APR program, right? It's always like a trick to no, it doesn't really matter. What's the fastest way to do this? Maybe what? It's gonna sign into my McGraw-Hill account. They certainly make it difficult. It's like, God forbid somebody ever accessed one of their videos without paying for the book. This is a good video, but again, it's always kind of like multiple steps to like actually get there. Gentle, make sure you guys are following along. Share. It's exhausting. So we go back to urinary video. Here's all the videos. Okay, here we go.
The micturition reflex is an autonomic reflex. It involves impulses traveling from the urinary bladder to the sacral region of the spinal cord.
So remember, autonomic means automatic without voluntary control. So this does tie into what we covered in the nervous system.
It is coordinated by neurons in the spinal cord and can be influenced by signals from the brain. When the urinary bladder becomes distended by the accumulation of urine, there is an increase in the frequency of action potentials carried from the bladder
Remember, action potentials are just nerve impulses.
Wall to the sacral region of the spinal cord. In response, parasympathetic neurons from the spinal cord to the bladder are stimulated. and this causes the smooth muscle of the bladder wall to contract.
Parasympathetic is rest and digest.
The sensory signals to the sacral region of the spinal cord also stimulate ascending pathways to the pons and cerebrum, which results in a conscious desire to urinate. If urination is not appropriate at the time, the brain sends impulses to the sacral region of the spinal cord to inhibit micturition. Impulses carried via somatic motor neurons keep the external urethral sphincter contracted, which also prevents urination. When urination is desired, signals from the brain stimulate the micturition reflex. To initiate urination, the brain decreases action potentials in the somatic motor neurons to relax the external urethral sphincter.
So it's saying the signals are causing the external urethral sphincter to remain contracted, right? And so a decrease in signals are then going to allow it to relax.
The micturition reflex is an autonomic.
The end. Okay. Sorry, but that was at the end. Okay, let's see if we can get back to urinary system. Okay, there we go. Right, so that's a micturition reflex. Okay, so the detrusor muscle is the muscle within the bladder. So that contracts, and the external urethral sphincter must also relax. Okay, so you can override the reflex and keep it contracted. It's the stretching of the bladder, right, that's going to send nerve impulses to trigger the reflex. So this is like one of the questions we had for lab homework, the difference between the male and female urethra. So the female urethra is much shorter. The opening is between the vagina and the clitoris. The male urethra being about 20 centimeters. And this serves as a pathway for the urinary as well as the reproductive system. And you can see the prostate gland surrounds the urethra at the base of the bladder, and then the urethra extends through the penis. So this is just showing a cross section of the urethra. So there are some drugs or compounds that increase the urine flow rate. So these you would call diuretics. So I remember like diuretic, like di or like diameter, so it goes like straight through. I don't know, that's how I remember it. Right. So caffeine and some drugs inhibit sodium reabsorption. So this is occurring in the distal convoluted tubule. Remember, water follows the salt. So if you're not reabsorbing as much sodium, you're not reabsorbing as much water. And alcohol inhibits the secretion of ADH. So I like from the video, ADH, a damn hangover. So being hungover due to like dehydration, which can cause, like that headachy feeling. Okay, so breaking the seal, is this myth or is this reality?
It is so soft,
You
and it
can get
is.
rid of
Here in this age of reason, there is still a place where superstition reigns supreme. Until today, I will bring light to the bar urinal, figuratively, because the darker a bar bathroom is in reality, the better I think it is for all of us. So you've probably heard of breaking the seal. It's the idea that if you're at a bar drinking, you can hold your pee almost indefinitely, but then the second you go to the bathroom, you are going to have to pee constantly for the rest of the night. I know people who swear by this, but I've always been skeptical. Thankfully, some urologists, who are probably also drunks, decided to figure out what's up. And what's up is that this just feels true. Here's what's going on. Alcohol suppresses vasopressin, which is an anti-diuretic hormone in your body. ADH regulates the water in your kidneys. So if you have a lot of ADH, your kidneys absorb more water. And if you have less of it, that water goes into the rest of your body, which means more water in your bladder. So you're in the bar, you're drinking a lot of fluid, and you're stopping your body from absorbing it, right? Alcohol is also a bladder irritant for a lot of people. So the more of it you drink, the more your bladder is going to feel uncomfortable and full, even when it's not. And these effects increase over time. The longer you add alcohol to your system, the more it suppresses that ADH, and it also makes your bladder feel more and more irritated. So if you've been holding it in for a long time, it can feel like you've broken some sort of seal, but really, these effects have all been building up anyway. By the time you initially go to the bathroom, they're all in their late stages. So it feels like you've skipped from not having to go to having to go all the time. But in reality, if you hadn't waited, if you weren't worried about the seal, and you just kind of peed gradually whenever you felt like you needed to, you actually would have wound up peeing the same amount. And you would have felt those effects building up slowly. It's all in your head, man, I am telling you. Well, it's more in your kidneys and bladder than your head. You get what I mean. What I mean is, do not worry about the seal. The seal has no power over you. Pee whenever you feel, assuming it is in an appropriate place. I will tell you one thing that makes you have to pee, and that is talking about peeing for 2 minutes straight. So I'm gonna go do that. You subscribe, and Lacey will be here today with another video. She could be in the bathroom.
Right, so breaking the seal is not a true phenomena. Make sure we're back on the right there. Go. Right, so no such thing breaking the seal. Right, so here are just two sets of Greek and Latin words. We have glomer and nephro and reno. So glomer is referring to the glomerulus, so it means little bowl. And then nephro in reno referring to kidney. They're just going to build two terms. Inflammation of the glomerulus of the nephron and pertaining to the kidneys. So this would be glomerulonephritis. So this is a phenomenon that typically occurs after you've had a strep throat, streptococcus infection, and you have strep antibody complexes that actually get trapped in the glomerular filter and cause irritation. And that leads to glomerulonephritis. Pertaining to kidneys, this one's easy, renal. Okay, renal. So the next thing I want to do is I want to look at the exam review sheet, which I just did some updates. This is Saturday morning. I just did some updates. So if you printed it out earlier, there's just a few changes. Let's check and make sure guys are on the right screen. This is weird because without anybody here, there's no one telling me if this is right or wrong. Should be right. Okay. All right. So yeah, this is posted in Blackboard. So the test is going to be 60 multiple choice questions. There is a small matching section on the word parts. There's about 20 questions on each of the units we covered, starting with the nervous system. I just reviewed, looked at the test, looked at the review sheet. Everything should be all set. Right. So going back to the beginning of the nervous system has two main components, the central and peripheral nervous system. Central, just think running down the middle, brain and spinal cord. Peripheral, anything extending from this area, cranial and spinal nerves. The autonomic nervous system is the part of the nervous system that controls all automatic or involuntary functions, such as your internal organs. And it has two divisions that both have nerves going to the same organs. So they kind of work together, and their effects are sometimes opposed to each other. So sympathetic is fight or flight responses. Parasympathetic is your rest and digest. Okay. There are 12 pairs of cranial nerves, and there's just 3. 2 of them we saw in lab. So number 1 is olfactory for smell, 2 is optic, and number 8 is vestibular cochlear. So don't worry about any of the other cranial nerves. These are the 3 that are sensory only. All the others are mixed. So they have some motor and some sensory function. Okay, you have 31 pairs of spinal nerves. Parts of the neuron we also did in lab. The cell body or soma, the dendrites or little antennas that receive stimulus. The axon transmits nerve impulses. So chemically, they kind of call these action potentials, but it's easier for us to just think of them as nerve impulses or nerve signals. Okay. In lab, we looked at a motor neuron, right? So motor neurons or interneurons are the most prevalent in the body, and they have the myelin sheath on the axon formed by the Schwann cells. So some of this was in your Blackboard learning module. There's 3 types of neurons. There's bipolar. I don't know. I remember this like you have 2, it says ears, nose, there should be a comma there. You have 2 eyes, 2 ears, 2 nostrils. That's your special senses. The unipolar, very small. They're detecting stimuli, for instance, from your skin. They're found in your joints, your muscles, your internal organs. The multipolar, these are the motor and interneurons. These are the most common, most prevalent. The interneurons, I always think of these as your thinking neurons. Their connections allow like thought processes. They're found in the brain. So often they are connector. They mean the spinal cord too, but often they're connector between the sensory and the motor neuron. Right? So also Blackboard your learning module were the types of neural glial cells. These are cells that support the neurons. Right? Schwann cell, looked at that in lab. It makes the nerves impulses be able to move more quickly. Okay, oligodendrocytes. These are the cells in the central nervous system in your brain that form the myelin sheath. So it's a little different than the Schwann cell. There's be multiple Schwann cells covering an axon. The oligodendrocytes, they look like a little octopus. So they're little extensions form myelin sheaths on a variety of different neurons. The ependymal cells, these help create and circulate the cerebral spinal fluid. The astrocytes, sometimes they call like astro, like star. They're kind of the star of the neuroglial cells. They form the blood-brain barrier that helps restrict the movement of certain types of molecules from entering into the brain. So they're protective, trying to protect the brain. And the neuroglial cells, they will destroy bacteria or any cellular debris. This could be like dead neurons within the central nervous system, right? So the oligodendrocytes, ependymal, astrocytes, and microglial cells are just found in the central nervous system. The Schwann cells being found in the peripheral nervous system. Right, so in lecture, we talked about the synapse. This is just like, means like snap. It's the area where 2 neurons connect and communicate with each other. And at the end of the axon is where you find synaptic vesicles, which store neurotransmitters, right? So neurotransmitters are chemical messengers. They're stored in the synapse. They're released. Okay, so in this neuron, they call it presynaptic. I like neuron 1 and neuron 2. It's a little easier. So when neuron 1 is triggered to send a nerve impulse, the nerve impulse goes down the bottom of the axon, triggers the release of the neurotransmitter. It diffuses across the synaptic cleft. That's just the little tiny space between our two neurons. And it binds to receptors on neuron 2, right? The neurons don't stay there indefinitely. They're recycled. Often they're just returned to the synapse for reuse. So they might be broken down and then go back, kind of get put back together, or they call that uptake. That's when they're removed, right? So excitatory nerve impulses bring a neuron closer to threshold. And so threshold is when it sends a signal, right? So dopamine is your feel-good neurotransmitter. When it's released, it helps target your reward motivation center, right? Norepinephrine, right, which is kind of under the adrenaline category, is a neurotransmitter that increases your heart rate. There's also inhibitory neurotransmitters, and they're going to decrease the likelihood of our neuron number 2 from sending a signal. So dopamine can also so you have the same neurotransmitter, kind of have a different function depending on where it's found in the body. So this helps to reduce motor activity, which kind of smooths out your emotions. So people that have Parkinson's disease, that have kind of jerky movements, it's due to a death of the cells in this area that control this type of motor activity. It's like the basal nuclei. So acetylcholine, this it acts to increase your heart rate. Right, so we looked at like serotonin as a neurotransmitter. That's a mood stabilizer. This reduced levels of serotonin are associated with people suffering from depression. GABA is a neurotransmitter that has a calming effect, helps control your stress, anxiety, and fear. Right, so we looked at the effects of drugs on neurotransmitters. So beta blockers, they decrease your heart activity, they reduce your blood pressure, they block the binding of norepinephrine. Atrophy, which is used in surgical emergencies to increase blood pressure, blocks the binding of acetylcholine. SSRI, selective serotonin reuptake inhibitors. This is used to treat depression because it helps to block the reuptake of serotonin. Right, looking at the more recreational drugs, cocaine, right, increases dopamine release, which is your feel-good neurotransmitter, and helps to block dopamine removal at the synapse. So more dopamine present means more, more like reward, feel good, sensations. Methamphetamine triggers the release of dopamine, serotonin, and adrenaline. Alcohol and Valium increase your dopamine release. Marijuana inhibits brain activity, increases dopamine release. So you see a lot of these are like targeting this feel-good dopamine neurotransmitter. And heroin, morphine decrease sensation of pain and also increase dopamine release. Right, we did structures of the spinal cord. Remember, the outer layer is the gray matter. Outer layer is the white matter. Inner layer looks like a butterfly is the gray matter. Found in the center is the central canal, and this contains cerebral spinal fluid, which is a clear filtrate of blood, which is nutritive. Oh, in the ventricles of your brain, it provides cushioning because your brain would kind of start to crush down on itself, but especially the two large lateral ventricles, they're full of fluid, so your brain is kind of like a raft floating on water. The meninges, remember there's 3 layers from outer to inner: dura mater, arachnoid, pia mater. Cerebrospinal fluid is found in the central canal, the ventricles of the brain, and also the subarachnoid space. Okay, we had the parts of the brain worksheet refer to that. The brain is divided by hemispheres, connected by corpus callosum. The lobes of the brain, right? This goes along the skull bones: frontal, parietal, occipital, temporal. The insula is kind of under the frontal lobe. So insula means like insulated, so it's kind of internal. And that's why it doesn't have an associated skull bone associated with it. Cerebrum, site of intelligence, personality, memory, learning. Your thalamus, just a relay station for sensory information. Cerebellum coordinates movement and balance. Okay, remember the tissue arrangements called the arbovites. The corpus callosum. I'm not sure. Connects. This is weird. I'm gonna change this. Connects the two cerebral. Something's missing here. Hemispheres. No, meninges. Okay, location of 3 layers. It's easy. Brain and spinal cord. Spinal cord. Hypothalamus, regulatory center for homeostasis. Pons links brain to the spinal cord, controls rate and depth of breathing. Medulla oblongata, vital processes, heart rate, blood pressure, and control certain reflexes. Okay, we looked at some reflex arcs. And 2, the withdrawal reflex if you put your hand on a hot stove or you step on a tack. This is to protect the body from painful stimulus, from tissue damage. The patellar reflex, this is the simplest reflex. There doesn't involve an interneuron. You hit your patellar tendon with the little hammer and your leg kicks forward. Okay, the digestive system, right? So digestion consists of 2 processes, mechanical and chemical. The layers of the alimentary canal, we're just going to focus on the mucosa. That's a hint. Innermost layer, that's where the food absorption occurs. Deep, we're moving outward to that, you'd have submucosa, muscularis, serosa. I'm congested today. All right, the mouth, or your oral cavity. Remember, your tongue has your taste receptors, the salivary glands initiating carbohydrate digestion. We looked at these in lab, the 3 pairs: parotid, submandibular, and sublingual salivary glands. Your food, when mixed with saliva, forms a ball called bolus. Your hard and soft palate separate your oral and nasal cavity, because maybe you have nasal pharynx or oropharynx, right? So it's going to help separate that. When you swallow, right, soft palate's going to elevate to seal off your nasal cavity. Your teeth initiate mechanical digestion, chewing, breaking down food particles into smaller components, right? So a lot of this we did in lab. The crown, that's the part above the gum line. The root is below the gum line. The pulp cavity, which then leads to your root canal, has blood vessels and nerves. Okay, here's our 3 regions of the pharynx. Right, Esophagus, food tube, doesn't have any digestive function other than connecting your mouth to your stomach. Your rugae, these are the folds in your stomach, allow for it to expand when you eat. The small intestine has 3 regions: the duodenum, the jejunum, which does most of your digestion and absorption of nutrients, and then the ileum, which then leads to the cecum. Remember, the mesenteries keep your small intestine suspended from your posterior body wall. Your large intestine, okay, so it stores, it forms feces, it doesn't absorb any nutrients. There are some bacteria there that do manufacture essential vitamins, okay, but it's not absorbing any digested carbohydrates, proteins, or lipids. They have, several mass movements a day by peristalsis. Structurally, it differs from the small intestine that it only has strips of muscle surrounding it, and this forms pouches called haustra. The tenia coli is the strip of muscle, right? So movement through the large intestine is much slower. It has 3 regions. It goes ascending, transverse, descending, right? And then you would have sigmoid, rectum, anus. Okay. Your feces contains bacteria, mucus, undigested food. I'm going to add shedded cells that kind of come off in other areas of the digestive tract. Right, your liver, the function. There were more functions that we covered in lecture, so we're just going to narrow it down a bit for the test. You have these individual hepatic globules where blood flows in, which is high in nutrients, and this is where metabolic processing occurs. The other digestive function is secreting bile, which helps emulsify fats, right? So mechanical digestion breaks the fats into smaller components or globs. Think of globs when you hear the word globules. And the bile keeps the globules from coming together or recoalescing. All right, your gallbladder is embedded in the posterior aspect of your liver. It stores and concentrates bile between meals. Peristalsis is the movement that propels food through the alimentary canal. We just saw this also functions in the urinary system, moving urine from the kidneys to the bladder. Segmentation is like a mixing type of movement that also assists in mechanical digestion. So segmentation is mixing, peristalsis is moving it forward. So we went over a bunch of different enzymes of the digestive system. These are just a kind of a summary. Amylase breaks down starch. It's found in your mouth, in your saliva, and released from the pancreas. Lipases break down fats. Chemically, fats are called lipids. That's mostly released from the pancreas. Nucleases break down nucleic acids, kind of a minor component of your diet. And proteases break down protein. So we did a little bit about pepsin and in the stomach. Or re-bracket my brackets now. No. All right, just in urinary system. Location the kidneys, they're not within the peritoneal cavity. And they're asymmetrical because of the liver, right? So the left lobe is depressed a little bit because the left lobe of the liver is larger. Well, this is hard to say. Right, so the left lobe is higher than the right due to asymmetry of the liver. All right, structure, the hilum is the indentation in the middle, the regions, right, pelvis, medulla, cortex. This is kind of listing them from, yeah, it listed from inside to outside. Just outside to inside be cortex, medulla, pelvis, right? The flow of the filtrate, once it goes into the collecting duct, it goes the minor calyx, major calyx, renal, pelvis, ureter. We mentioned EPO stimulates red blood cell production. Nephrons, these are the microscopic functional units of the kidneys. Right? Some of this was in the Blackboard learning module. We kind of reviewed it in today's lecture. The renal corpuscle is the glomerulus, glomerular capsule. I don't know if you ever studied anatomy previously, the term that was used was Bowman's capsule. Now we got a lot of glomerular words going on. So it does the process of filtration, okay, removing small substances. You generate about 180 liters of filtrate a day. The nephron loop, okay, so proximal tubule reabsorbs glucose, amino acids, and water, does secrete additional substances to the filtrate. Examples: penicillin. Nephron loop, also called renal loop, goes down, curves and up. So it's got descending limb, ascending limb. So just the key here, this is only water reabsorption. Distal tubule, no water reabsorption unless acted on by ADH or aldosterone. So ADH is that's when your hypothalamus detects that your blood water volume is low, and aldosterone is released when your blood pressure drops, both causing more water reabsorption. Right, so urine, we said it's 95% water, 95%, that's easy to remember, right? 95% water. Urea, uric acid, creatinine, it's normally yellow due to the presence of urochrome. Make sure you know what should not be in your urine: proteins, glucose, ketones, blood cells. The bladder has 3 openings at the bottom: the trigone, 2 ureters, and 1 urethra. The volume held can vary between 200 and 800 milliliters. The urethra, the function, it's transporting the urine from the bladder to outside the body. It's longer in males than females. We did the micturition, which just means urination reflex. So stretch receptors in the bladder that start to detect the bladder's full, and then this initiates the reflex, which you can override, right, to an extent. When urination occurs, this muscle in the bladder contracts, and then the external and internal urethral muscles must relax. All right, so aldosterone from the adrenal gland makes, so DCT for distal convolute tubule, more permeable to water, increases blood volume, blood pressure. This is due to sodium ion reabsorption. Don't worry about that. ADH by hypothalamus. It's released by pituitary gland. Don't worry about posterior. There's different functions in different areas. It also makes the distal convolute tubule more permeable to water. Right, so they have aldosterone and ADH have the same outcome. They're just triggered differently. Aldosterone by decrease in blood pressure, ADH by detected in decrease in water concentration of your blood, okay, or increase in solute concentration, right? EPO increases red blood cell production. Your diuretics increase urine production. Some compounds decrease water reabsorption, and alcohol decreases the release of ADH. Okay, so just make sure you go over the Greek and Latin word parts. Just last thing I'm going to do. Sorry, I'm scrolling. Yeah, so you'll find the review. Sometimes you follow along with what I'm selecting, and sometimes you just don't. There's also a link in Blackboard that has a Kahoot review. If you want to try that, you can use that also to review the information, make sure that you go through the Unit 3 review sheet that I just covered. So hopefully you get a chance to go through all of this before Thursday, so that if you have any questions, you're able to ask me in lab. Okay, everybody enjoys your weekend. I'm going to post this as soon as I get the recording to the cloud.